We're going to finish up this week with a review of some of the microscopic structures, cellular structures that we did before. And I'm going to use the same slides that I used before. So if you feel like you're in good shape with this, you can skip this part of the videos. That's why I separated this into a separate part, so that if you felt like you didn't really need to go through this again, you didn't have to. So from this diagram, first of all, we're looking at these two structures right here. And if you recall, these are the mitochondria. The mitochondria are important in the generation of energy, and they convert nutrients into energy, and this energy is used for the various cellular functions that are required. Mitochondria are important for active transport and various other motilities and things that have to occur that require energy for them to recur. The second structures are shown here. They're thick-walled, as you can see. These are the lysosomes. The lysosomes are organelles that are responsible for the breakdown of molecules. They break down proteins, carbohydrates, and lipids. The third structure are these little structures. Some are associated with membranes, others are not. These are called ribosomes. Ribosomes are organelles that are responsible for protein synthesis. Here's another diagram. We'll look at some other structures in this diagram. The endoplasmic reticulum. The endoplasmic reticulum is shown here and here. Some endoplasmic reticulum has ribosomes associated with it, and that's called the rough endoplasmic reticulum and is used to make proteins. Other endoplasmic reticulum here has no ribosomes associated with it. That's called the smooth endoplasmic reticulum, and it is used to make other types of chemicals. This is the rough ER here and the smooth ER here. This apparatus right here is what some people have called the stack of pancakes. This is called the Golgi apparatus. The Golgi apparatus, or the Golgi body, is an organelle where proteins and lipids are stored and then modified to suit cellular means depending on what the cells need. Below that, and here these are almost always in a sort of right angle to each other, are the centrioles. Centrioles are used to form flagella, cilia, and then you'll remember in our study of mitosis and meiosis, they are also important in forming the microtubules that pull the chromosomes apart. Also within the cell are spiral strands of proteins that form tubules known as microtubules, and as I said, those function to move things through the cell, not the least of which is the chromosomes during cellular reproduction. Now, in terms of how cells take in materials or get rid of materials, there are a couple processes shown in this slide. One is known as phagocytosis. Phagocytosis is shown on the left here. It is a process where a cell engulfs the foreign substance and then forms a vacuole around it. And many times a lysosome will come up and dump digestive enzymes into that vacuole, and then the material is then digested. If the material is more water-soluble or liquid, then that process is known as the penocytosis. Now what you can see here, the first vacuole that I was talking about was called a phagocytic vacuole, and it is a organ that holds whatever is engulfed, whatever large particle is engulfed. A penocytotic vesicle is a vesicle that forms around the fluid material, and many times this fluid material will hold a larger molecule, not large enough to be considered phagocytosis. And then the reverse process occurs when the vacuole, then now known as the secretion vacuole or vesicle, will then expel its material into the external environment. The nucleus is made up of several structures shown in this diagram here. First of all, the nucleus itself is where the DNA is stored and replicated, and it's stored in the form of chromosomes and histones, which are proteins that are associated with the chromosomes. And on the outside of the nucleus is what's known as the nuclear membrane. This membrane is quite porous. It separates the nucleus from the cytoplasm, but it has many holes that allow for the movement of RNA and in and out of the nucleus, and allow for the transport of things such as RNA. Chromatin is just another term for clusters of associated DNA, RNA, and proteins in the nucleus. And then this large body here and here is called the nucleolus, and the nucleolus is the center of RNA synthesis. And you'll remember that RNA is synthesized from a DNA template in the process of transcription. Now our last diagram is shown here, shows the microscopic cellular skeletal structure, if you will. And this shows this sort of girder-like structure known as the cytoskeleton. This is a network of fibers that holds the cell together, giving cell its shape, and allows for the movements of materials throughout the cell. Individual parts of the cytoskeleton are known as microfilaments. These are fine thread-like proteins that are found throughout here that perform the function that I've stated before, that is the movement of materials through the cell and holding the cell in its shape. Intermediate filaments are thread-like proteins in the cell's cytoskeleton that are roughly twice as thick as microfilaments, and they sort of associate the microfilaments with each other. And so they sort of, if you think of the microfilaments as long girders, then the intermediate filaments hold the girders together. If the girders are in one direction vertically, then the intermediate filaments are horizontal, holding them together and giving you a very solid structure. Now when we talk about the transportation of materials, we remember that the important outer part of the cell is known as the cellular membrane. And cellular membranes are made up of phospholipids. Phospholipids has one end that's a fatty acid polar part of the molecule, and the other end that has a long hydrophobic tail. A phospholipid is a lipid where the acid here is substituted with a phosphate. So you have this polar area in this area and these long hydrophobic tails. And then here's glycerol. That's why it's triglycerides that are phosphated. And what you end up having is a situation where the heads all associate with each other because of chemical likeness. The tails project in toward each other, and then the heads on the inside do the same thing, and this forms an almost impenetrable barrier to the outside. The head part is hydrophilic, meaning that it loves water. It's polar. Water-soluble materials can dissolve in this area. And then the hydrophobic area, which is in here, does not allow water or water-soluble materials to enter. Now here's the plasma membrane structure, and you remember this is a phospholipid bilayer. We showed that in the previous slide. You have a layer of lipid here, a layer of lipid here, so it's a bilayer that's moderately impenetrable. But then you have channels that allow for things to cross. Some of these channels are made up of glycoproteins, which are carbohydrates associated with proteins that allow for things to cross. In this case, this glycoprotein is functioning as a receptor, out in the external part that can be detected by something else. And then you have glycolipid, which is a similar type of receptor that is a carbohydrate associated with a lipid. But the plasma membrane of cells in the body have channels that allow for things to pass through, but they selectively allow the passage of various materials. Now, when you talk about the transportation of materials, materials are transported generally one of two ways, passively and actively. Passive transport is the movement of molecules through the plasma membrane according to the laws of physics and the laws of diffusion and osmosis, which means that material tends to flow from high concentration to low concentration to try to balance the concentration, from areas of low osmotic pressure, or from high osmotic pressure to lower osmotic pressure to balance the osmotic pressure. There is no expenditure of energy in passive transportation. Active transportation is the movement of molecules through a plasma membrane, which requires energy, and it almost always occurs against the laws of physics, the laws of diffusion, and the laws of osmosis. So generally the energy is expended in order to overcome the natural law that's being overcome by the act of transportation. So when you're transporting materials according to the laws of diffusion, you can have one of three types of solutions: hypotonic, hypertonic, or isotonic. An isotonic solution, which is shown in the middle here, is a solution where the concentration of the solutes, pictured as these little dots, is essentially equal within and without the cell, so that the membrane itself on either side of the membrane is approximately the same amount of solutes. There tends to be motion of molecules across, but the concentration does not change during passive transportation. Hypertonic solutions, shown on the right, is a situation where outside the cell the concentration of solutes is greater than inside the cell. And because of that, the natural tendency in passive transport would tend to transport material into the cell. If you want to keep this situation the way it is, you have to expend energy. And there are situations where there is active pumping of material out of the cell. Sodium, for example, is pumped out of the cell so that the concentration of sodium outside the cell is greater than the concentration of sodium inside the cell. So it's hypertonic with respect to sodium, and that is maintained by the act of transport. Conversely, hypotonic solutions are solutions where the solutes on the outside are less than the solutes on the inside. And in this case, fluid would tend to naturally flow into the cell to try to dilute the inside of the cell. Now, because that occurs, now this is a situation that can occur with potassium. The concentration of potassium outside the cell is commonly far less than the concentration of potassium inside the cell. And this is achieved by the active pumping of potassium inside the cell, while the pumping of sodium is being done outside the cell. And this can result in any of these following cells. You can have a hypertonic cell, an isotonic cell, or a hypotonic cell. If you have a hypertonic cell, then plasmolysis can occur. Plasmolysis is a situation where a walled cell membrane collapses due to the lack of water. The water is moved out of the cell in an attempt to exchange the amount of solutes, and basically the cell collapses on itself. Conversely, on the right here, you have cytolysis, which is the rupturing the cell due to an increased amount of fluid entering into the cell.